Welcome. This is KCSE 2013 Biology Paper 3. Let's see what you are supposed to do. In question 1, the photograph below shows the inner surface of the upper left side of the rib cage. So you can see this is the rib cage, these are the ribs. Then you are told, name the bone covered by the fatty tissue labeled K. Name the bone. This one now, this is the bone that is covered by the fatty tissue labeled K. Now that is the sternum. Then you are told, explain the role of the part labeled M in inhalation. Part labeled M, these are the ribs. M is the rib. Now you are told to explain the role during inhalation. You will say, after the internal intercostal muscle relax, after the internal let's write here this question Roman 2, and after the internal intercostal muscles relaxes, the rib cage moves upwards and outwards. After this one increases the temperature and decreases the pressure. This increases the volume, not the temperature, sorry. Increases the volume and decreases the pressure. The pressure in the rib cage. The rib cage, we have said that is the rib cage M. Now after the pressure and the volume have the pressure has decreased and the volume have increased, now the air is forced into the lung. The air is forced into the lungs. That is the role of the part labeled M. Now question B, you are told the photograph the mammalian vertebrae. Now you should be able to identify the mammalian vertebrae. This is the thoracic because of the long neural spine and the centrum, which is broad wide neural canal, and the transverse process. Now this is the thoracic vertebrae. Now you are told, state the view of the vertebra presented. Now this is the dorsal view or the anterior view. The anterior should be able to know the difference between anterior and posterior. As in the early video we said, the posterior, this is the downwards, this is the lower part, while the anterior, this is the upper part. Should be able to know the differences. Now this is the anterior view or the dorsal view. The anterior is seeing the upper part, we are not seeing the lower part. This is now the anterior view or the dorsal view. Then Roman 2, you are told name and state one function of the part labeled T. Part labeled T is the neural canal. This is it is pointing the center neural canal. That is the neural canal. Now you say name, this is the neural canal, and therefore this allows for the passage of the spinal cord. Yeah. Then you are told Roman 3, how are the parts labeled S and V adapted to their functions? Now S and V, S is the neural spine, and then V, this is the centrum. Now the adaptation of these two, you will say S being the neural spine is long, that is how it is adapted. Now function is to provide surface for muscle attachment. Yes. Now and then in V, which is the centrum, it is thick. That is how it is adapted. It is thick or it is broad to bear the weight of the body. Now question 2, you are provided with a food sample labeled C. Using the reagent provided, carry out the test and identify the food substance present in the sample. Now our space is very small, but we have big space. Now you are required to test for reducing sugar. Now in the procedure will say put 2 centimeters cubed or 2 millimeter of the food substance C in a test tube, and then add equal amount of Benedict solution, and then heat to boil. Either in a water bath, you can write in a hot water bath, or heat to boil. Now the observation were the blue color of Benedict's solution remain blue. It was the observation. Now in conclusion, there was absence of reducing sugars absent. Now we are testing for non-reducing sugar. In this, the procedure is very big. You should be very careful while writing this procedure. Now we'll start saying put 2 ml of C in a test tube, and then add few drops of dilute hydrochloric acid, add few drops of dilute hydrochloric acid, then you place the test tube in hot water in a hot water bath for about 3 minutes, and then remove the test tube and cool with cold water. Then cool with cold water. Then add sodium hydrogen carbonate drop by drop until fizzing stops. Then add Benedict solution. Then heat to boil or warm in a water bath. Then heat to boil. Now here the color changed to yellow, then or orange or brown. That is the color you expected. Now conclusion, the non-reducing present, and reducing sugars present. Now for procedure 3, for test for proteins. Now you are required to put 2 ml of the food substance in a test tube. Then add sodium hydroxide equal amount and shake. Then you shake. Then add copper 2 sulfate drop by drop. Then shake. Then you observe the color. Here the color changed. To purple. Therefore, proteins were present. Question 3. Below are some photographs showing some observable features of leaves. Then you are instructed by being told, using the features in the order given below, construct a dichotomous key that can be used to identify the specimens. Now the first one, you should start with leaf simple or leaf compound, then leaf venation, margin, arrangement of leaves, pinnate or trifoliate. Now let's start with the first one, leaf simple and leaf compound. You should be able to identify the leaf simples and leaf compound. So this is the leaf compound, then this is leaf compound, and even this one, this is compound, and there this is simple. Now let's start by constructing. You say 1A leaf simple, and then B leaf compound. Then we start with the first one, the leaf simple. We have said there are 3. Now you cannot write all the names. You should now look for a characteristics that will differentiate the 3. Now you will say here, because you don't have any only one, there are many, you write go to 2, and then you go to 2a. Now you look the 3, how you can differentiate an observable feature. Now I can see parallel veined leaf, network veined leaves. Now you say leaf with network veins. Now you say leaf net veined, this is a short form, net veined, and then B, leaf with parallel veins. Now with the network veins there are 2, but with parallel veins is 1. Now we write this one, we write the name of the leaf, which is the Commelinaceae. That is a scientific name, Commelinaceae. Now here, because there are 2, you say go to 3. Then you differentiate. I can see the leaf with smooth margin and leaf with serrated margin. Now you have differentiate them. 3a, you say leaf with serrated margin, and then B, leaf with smooth margin. Then you identify there are only 2, the Malvaceae and the Nystaginae. Now you write their names. Here this is the Malvaceae, and here this is the Nystaginaceae. Then we have finished with the simple leaf. Now we go back to the A, 1b. Go back to 1b, which is compound leaf. Now we have finished with the leaf simple. Now here we say go to 4. Then we write 4a. Let's see the compound leaves. Now here we will say leaf with leaf opposite, that have the leaves opposite, and the other one with alternate. You'll say leaf opposite and leaf alternate. These are the opposite and these are alternating. Now you'll say even this is opposite and this is alternating. Only one is alternating, which one is this one, but these 2 are opposite. Now you will say leaves with leaf opposite, and B, leaf alternate. Leaf alternate. We have seen leaf alternate is only 1, but opposite there are 2. Therefore, we will say leaf alternate, its name is this one, which is Bingolanaceae. While here now, because there are 2, we say go to 5. Now in 5, say 5a and b. This is simple because they are the only 2. Now this we will say, because there are 3, we'll say leaf trifoliate, and this one there are many, leaf pinnate. I will say leaf trifoliate, and then leaf pinnate. Now automatically you write the names where the leaf trifoliate is the compositae, the compositae, and the other one is papilionaceae. Papilionaceae. Now that's all. Thank you to our viewers. I'll urge you to subscribe, share and comment, and God will bless you. This work is not very easy, but do us a favor, please subscribe.